Okay, so before starting the talk, I'd like to firstly explain the scope of this symposium. Actually, this is a kickoff project of our 5-year group grant supported by the MEX of Japanese government. And I'm the project leader of this group, and this is named Transformative Research Area. And the name of the project is Emergence of Brain Function from the Dynamic Connectome. And here are co-PIs listed here. And this is the very first year of this project. So we plan this symposium to discuss our research aims, as well as to facilitate the collaboration. So before starting this symposium, I'd like to introduce what we are thinking now and what we want to do in the next 5 years. Okay, so during the past decade or so, a lot of study has identified the kind of cell types that constitute each of the organ, but that kind of information tells us a lot about the molecular interaction among different kinds of cells. But in the case of the brain, there is another layer of intercellular communication, that is wiring. So we really need to understand the wiring diagram of the neuronal circuits. So that is why many people are now trying to understand the wiring diagram of the neuronal circuits. And there are several connectomics projects are ongoing, but we think that even if we fully understand the wiring pattern of the brain, we probably still don't fully understand how the brain works. That is partly because some of the brain functions are kind of emergent phenomena. That means that the behavior of the population may not really just the sum of each component. For example, each of the neurons receives a lot of synaptic inputs, but it's not always just the sum of the synaptic inputs. Sometimes there is a non-linear transformation happening. And that is also the case for the neuronal circuits. It's not just the sum of the behavior of individual neurons. So we probably need to understand what is the underlying principle of that kind of emergent properties of the neurons or neuronal circuits. So we want to understand what is the principle that control this kind of behaviors. And also another interesting point here is that our brain function is not complete from the beginning. Actually, most of the brain functions are acquired during the changes of the structures. And that is well known in the developmental studies. But this is an example of the LTP or LPD. Upon some stimulus, the connectivity will change, and that is a kind of basis for the elemental process. But actually, we still really understand what kind of network changes are happening and what kind of structural changes as a whole give rise to the new functions of the neural circuits. So we want to understand this kind of system level changes and underlying logics. So so in this research project, we want to understand the basis of the brain functions based on the dynamic connectome. So how these structural changes and what kind of changes lead to the functional changes. Currently, the ongoing connectomics strategy trying to understand the basis of the brain function based on the kind of static connectome informations. But what we want to do in the next 5 years would be to understand the basis of the brain function based on the dynamic changes of the structures. So for this purpose, we probably have to understand the connectome or the structures of the circuits at the multiple time points or in different animals or different conditions. Also, we have to combine the structural imaging with the in vivo imaging. And also, we have to combine the structural imaging and the functional imaging. So during the combination of this kind of strategy, we want to understand the principles that orchestrate this kind of emergent functional properties. Here, I'd like to give some example from our studies. This is an example from our laboratory. During development, it is well known that the receptor fields of the neurons are gradually tuned. For example, this is an example of the microcylinder factor, and these neurons initially extend multiple dendrites. So that means that they receive a heterogeneous kind of sensory inputs. But during development, they prune the unnecessary connectivity, and eventually they establish just one type of dendrite. So eventually their receptor field will be tuned to just one type of autoreceptors. And our recent study have shown that this is ensured by the intrasynaptic communication. We recently showed that the synaptic input to just one type of dendrite send a lateral inhibition signal that facilitates the pruning of the weaker dendrites. So there is some kind of communication across the synapses. So so that means that this is not just an ATP or ATT in individual synapses. It's an interaction among all kinds of synapses. So that means that one lesson here is that we probably need to study many synapses or all synapses simultaneously to understand how the different components communicate to each other. The next example, this is from a recent study from Goda Sensei's group. This is showing the distribution of the synaptic strengths. And this means that the distribution of the synaptic strength is highly skewed. And so, but if the distribution is normally distributed, it's not so efficient actually. Simulation indicates that the skewed distribution is quite useful for the learning process. And on the right side, there is another example from Murayama-san's group. He studied the recorded a lot of neurons from the cortex and found that there are very skewed connectivity in the cortex as well. And so this is a totally different scale of phenomenon, but seems to indicate that there are kind of common principles that is critical for the brain functions. So that may be that the bias distribution of the distribution might be quite useful for the stabilizing specific circuits or the conditions. That may be the key to understand the emergent functions. Okay, this is another example. This is from Kubota Sensei's group. Sometimes the emergent function that leads to the behavior can be stepwise. And he discovered that during the motor learning process, there are two different steps of plasticity. In the early stage of plasticity, the cortical inputs play a major role. But at the later stage, the thalamic input play a more important role. So this is just one example, but in many cases, the behavior is established through the stepwise emergent changes. Okay,